Just going to share my screen with you so that you can see the PowerPoint. So this one's pretty short. You can see only 24 slides. So we're going to start at the beginning here. This is the first unit, Chemistry of Life. This is geared toward the AP exam, and then some information that will help you with the ECE exam as well, and give us a nice foundation when we start botany. So we still have botany and taxonomy left. Okay. So structure of water and hydrogen bonds is going to be important. We're going to go through the organic macromolecules and then nucleic acids. There's a heavy emphasis on the curriculum and exam description on nucleic acids. So we'll go through them, but we'll also cover them when we do molecular genetics. All right. And then the science practices, the three main ones here is to describe the characteristics of biological concepts, processes, or models, represented visually. Okay. And then you can also see there's another one that predict cause or effect using visual representations. So you've done this using the POGLs that we have done over the last year. Okay, we've really worked on that skill. Properties of water, if you remember, water is a polar covalent bond. So the electrons are shared between oxygen and hydrogen, but not equally. Oxygen is highly electronegative, so the electrons spend most of their time with oxygen and very little time with hydrogen. And as a result, our oxygen molecule here has a negative polarity of 2, and the hydrogen has a positive polarity here. And this allows them to form hydrogen bonds. These hydrogen bonds are weak and they're temporary because these electrons that are in oxygen have to go back to hydrogen. And when that happens, these hydrogen bonds break. But water can form up to 4 hydrogen bonds. And that property of water, due to its structure, really allows for life on Earth. Okay, so the hydrogen bonds between the water molecule, these result in cohesion, adhesion, and surface tension. Cohesion, if you look down here, that's the water molecule to water molecule. So this hydrogen here feels positive, the oxygen feels negative, we have a hydrogen bond. Adhesion is water to xylem, okay, or another substance. So it's water to another molecule. And this is very important, cohesion with adhesion, that is capillary action, and that's what allows for transpirational pull. So water is going to be evaporated from the stomata of the leaf, and that creates a negative pressure, a negative pull, which decreases the water potential. So the water wants to go up from roots through the leaves and evaporate. And that allows all parts of the plants to get water. The other thing we see is surface tension. So you can see here the little basilisk lizard. You see this if you get that little, like, if you overfill your cup with water and you get that little, like, top of it that goes over and it doesn't break, okay? And some fish take advantage of this to actually have their eggs on the top of the water. It's using that surface tension. Water is also less dense as a solid because it creates maximum amount of hydrogen bonds that you see here. So it has 4 hydrogen bonds. That allows a lot of space in between the water molecules. That lattice structure, more air pockets, and so it flows. It's important to life. One, if it was more dense, we'd just be a solid ice here on Earth. But also, it helps insulate the water below, so it doesn't get below 4 degrees Celsius, which is why even though ectotherms can't live on land in super cold places like the poles, they can live in the water because it doesn't get too cold for them to survive. They also have hydrophilic and hydrophobic. So hydrophobic, those are going to be your fats or lipids. So waxes, your steroid-based hormones, cholesterol, your triglycerides. And they can't dissolve in water because they're nonpolar. I have covalent bonds where the electrons are being shared equally. A lot of times these are hydrocarbons. Hydrophilic can dissolve in water because they're either charged or they're polar. So anything with an ionic bond or a polar covalent bond. The fact that hydrophilic solutes can dissolve in water is why water is an excellent solvent. And that's why, for instance, you look at your blood, it has a lot of water in it, right? Because you need to transport sodium ions, potassium ions, right? You need to transport food. So all of that can be easily transported through your blood because a good percent of your blood is water, and it's a good solvent for that. Has a high specific heat. That's the amount of energy that you need to change the 1 gram of water or 1 milliliter of water, 1 degree Celsius. And it has to absorb lots of energy to do that because you have to break all those hydrogen bonds. It's important for moderating our temperatures. It also helps moderate the Earth's temperature. So if we're looking at the news right now, you'll notice that if you're inland in Connecticut, when it's like 58, 60 degrees, it's still cooler down by the coast because the water's temperature at the coast hasn't changed, right? And so that's causing them to have a cooler temperature. High heat evaporation, this is the amount of energy needed to go from a liquid to a gas. And again, you have to break all those hydrogen bonds to change the state. This cools organisms, evaporative cooling. So here's your skin, right? And the sweat here is going to evaporate as long as it's not too humid out. And as it goes from that liquid to a gas, the energy to do that is coming from the heat on your skin. So that's why it cools you. That's why when you get out of the pool, you're suddenly very cool. Organic compounds all contain carbon. Now, not everything that contains carbon is organic. Carbon monoxide is not organic, and neither is carbon dioxide. But when we're looking at our 4 major macromolecules, biological macromolecules, which are your proteins, your lipids, your carbohydrates, and your nucleic acids, they all contain carbon. And that's because carbon can make the most covalent bonds 4, and it is able, because it doesn't have this huge nucleus, it's able to make these really neat structures, right? It can have some branching, circles, all kinds of things. And if I was just to look at the carbon with the hydrogen, it would be hydrophobic. So how does it get to be hydrophilic? You're mainly water, so you have to change that. And that's done by the functional groups. So in your book, which I'm looking over here so I can give you the page number, your functional groups are on page 43 of your book. So we have our hydroxyl group. We're going to find that alcohols. That's going to make things, that's going to be a polar covalent bond. So now it can mix in water. Okay, and we're going to find that in, for instance, carbohydrates. We have our carbonyl, and we all have our carboxyl. We'll see the carboxyl group in our amino acids. That's the acid part. The amino part is the amino group, which acts as a base, which is NH3. You have your phosphate group. That's found in your nucleic acids. It's also going to be found in your phospholipid bilayer. It's important for ATP. That's your energy transfer. You have your sulfohydryl group that is in some of your amino acids that creates a disulfide bond, a nice strong bond. And then if you see methyl group, that's a hydrophobic group. That's CH3. Okay. So organic macromolecules, they all have carbon, but we're going to see a few other elements that differ. Nitrogen, you can see nitrogen here. Here's that amino group, NH3. It's gonna, and then we see our amino group down here in our nucleic acids. And remember, ATP is a modified nucleic acid. So if I have a problem that says, for instance, if there's low nitrogen in the soil, what is the plant not going to be able to make? And then they're not going to tell you proteins, nucleic acids, they usually will have a graph or something else you have to look at so they can put a science practice in there, but you would know, oh, proteins and nucleic acids. If it was sulfur, sulfur is only found in some of the amino acids. So sulfur is unique for amino acids. I can tell a protein and nucleic acid apart because phosphorus is found in nucleic acids, phosphate. And here we have some lipids, your phospholipids specifically, that's used to make membranes. It has that phosphate heads replace one of the 3 fatty acids. So this is going to have both a hydrophilic section and a hydrophobic section. Carbohydrates, they are going to be carbon, hydrogen, and oxygen. And the ratio is like 1 carbon to basically a water molecule, and that's where its name comes. Its bond is a glycosidic linkage, and that is formed by dehydration synthesis. So you dehydrate, you move water to make a bond, to make something synthesized. That can be broken by the opposite, which is hydrolysis, adding water, hydrolyze to break. And that's how all of the monomers of all these different organic macromolecules come together. Dehydration synthesis, and it's also how they all break, hydrolysis. Their function here is can be energy storage. That's glycogen in animals. And we store that in our skeletal muscles in our liver. It's about 4 kilocalories per gram. And starch in plants. Structurally, cellulose plant cell walls. And we can't break that down because structurally cellulose is a little bit different, the type of bond between those glucose molecules, right? And you may remember this, it's on page 47. You can look at the difference of the starch versus the glycogen and how that causes, we can break down starch, but we can't break down, we can break down starch, we can break down glycogen, but we can't break down cellulose. And you can see all 3 how they're different because glycogen and starch, their bonds with the oxygen is always down here, okay, in that glycosidic linkage. Whereas with our cellulose, it alternates up and down, okay, that facing. It's really good to look at that picture there, okay. I could not find that picture to post here, but that change in structure completely alters the function of the carbohydrates, even though they're all made of these monosaccharides. Proteins, these are monomers and amino acids. So here's your amino group, here's your carboxyl group. This is your central carbon, here's your hydrogen. The only part that changes is the residual group, that variable region, okay? And it's important because that helps create the tertiary structure of a protein. It's the tertiary structure of the protein that starts to be 3D. And all proteins have to get to that level, at least a function. Most will also get to quaternary, and we'll go over those in a minute. But if I have a mutation and get a different amino acid, and its R group behaves differently, my protein might not function. And we talked about that before with sickle cell. The bond here is a peptide bond, again, formed by dehydration synthesis, the covalent bond, and it's broken by hydrolysis. And the proteins do a lot of jobs. They can be structural, like keratin in your nails, enzymes, muscle, your muscle tissue is made of actin and myosin, antibodies, and your hydrophilic hormones like insulin. Okay. And we're going to see that the bond is always between the carboxyl. So here's where the carboxyl group was. And what came off that carboxyl was the hydroxyl, OH. So you can see over here, it still has the OH. And here's the amino group. And what came off that was the hydrogen, H, right? So H2O, water, basically. All right. Our primary structure, that's covalent bonds. Secondary structure, that is due to hydrogen bonds. So you have your alpha helix and, oops, sometimes it does that, and your beta pleated sheet. Okay, so these are happening between amino acids that are not right next to each other. Then I am going to also have tertiary. This is our R group interaction. So these can be hydrophobic, sorry again. They could be ionic bonds. They can be disulfide bridges. This is the level it has to get to because now it looks 3D, okay? And then quaternary is when you have more than one amino acid. So collagen would be a good example, or your hemoglobin has 4 units. When a protein denatures, it's losing those bonds, the quaternary, the tertiary, the secondary, and it's getting down to that primary structure, which does not have a 3D structure, so the protein can't function. Structure defines function. Lipids don't have a monomer. Their bond is called an ester bond. Again, formed by dehydration synthesis, broken by hydrolysis. It's a covalent bond. All of them are here. These can be, there's 2 types. We look at our triglycerides. Those are formed from a glycerol and 3 fatty acids, right? And in a phospholipid, one of these fatty acids replaced with a phosphate head. And the difference in these fatty acids are going to change the structure and therefore the function. And then we have our hydrophobic hormones. They're steroid-based. So steroids, 4 carbon rings. Cholesterol is a steroid, but also estrogen, testosterone, and progesterone. Those are steroid hormones. Aldosterone, if you remember, that's made in the adrenal glands. That's going to cause you to reabsorb more water, but it also can be a long-term stress response leading to high blood pressure. And cortisol, which causes you to store, so also made by the adrenal glands and adrenal cortex, causes you to store more fat around your middle. Again, it's a hormone of high stress, right? These, unlike hydrophilic hormones, estrogen, testosterone, progesterone, etc., these can get through the phospholipid bilayer, and their receptor will be inside the cell. Hydrophilic protein-based ones, it's outside. So you may recall that. We'll go into that a little more when we do cell communication. Here we have our phospholipid. So all your cell membranes are made of this phospholipid bilayer. All of your organelles that have membranes, so that's pretty much all of the organelles with the exception of ribosomes, also have this phospholipid bilayer. So it has the phosphate here, that's that hydrophilic head, and then 2 fatty acids. So it has double properties. The phosphate heads are going to be facing, if I was looking at a phospholipid bilayer of a cell, it's going to be facing the interstitial fluid, and this would be facing the cytosol. And the fatty acids just want to get away from anything that is hydrophilic, so they just are attracted to each other. And in between is the cholesterol here. Okay. And these are not bonded together, it's just a hydrophobic interaction. So this is just going to kind of float and they're moving. A lot of flexibility. We'll see the same structure in an organelle like the smoothie R or the rough ER. Nucleic acids, their monomer is a nucleotide. So if you remember, that's a sugar, a phosphate group, and a nitrogen base. The polymers are DNA and RNA. So here's your DNA here, it's double-stranded. Here's your RNA. The first one to evolve was actually RNA, was our first genetic material. That's the RNA world hypothesis, because RNA can get 3D structures. It can act as an enzyme, but it does mutate more rapidly. And that's why, with the exception of a few things like retroviruses, living things, anything that is on the cellular level, is going to be using DNA as its information storage, which is the function of our nucleic acid. And that's stored, remember, that information in your sequence of nitrogen bases. So DNA is double-stranded, RNA is single-stranded. DNA is thymine, RNA is uracil. DNA has deoxyribose, RNA has ribose. Ribose and uracil are less stable than deoxyribose and thymine, all right? And that's why RNA mutates more rapidly. Your purines, so they have double rings, that's adenine and guanine, right? And your pyrimidine, single ring, that's uracil, thymine, and cytosine. So adenine pairs with thymine in DNA and uracil in RNA. It has 2 hydrogen bonds between them, and we tend to find in DNA areas that are euchromatic, meaning they code for proteins, tend to be adenine and thymine rich. That doesn't mean they don't have guanine and cytosine, they just have more adenine and thymine. And then guanine and cytosine has 3 hydrogen bonds, and we find the heterochromatin, the non-coding regions, tend to be guanine and cytosine rich. Again, doesn't mean it doesn't have adenine and thymine, it's just more rich in guanine and cytosine. Again, structure, X, defining function. I need to get at that material, if it's a coding region, to make the mRNA and then eventually make the protein. But if it's a non-coding region, I don't need to get at that material as much, so it's better for it to be more stable with 3 hydrogen bonds. This was very much emphasized in the curriculum and exam description that's online. So that's the directionality of these nucleic acids. So when they're talking about 5' versus 3', remember they're referring to the carbons. So you start from the oxygen and you go clockwise. So 1, 2, 3, 4, 5. The 5th carbon always has a phosphate attached. Here we go again, starts the oxygen clockwise, 1, 2, 3. 3rd carbon always has hydroxyl attached. So DNA has one side that goes 5 to 3, the other 3 to 5. They're anti-parallel. It's important, right? If you remember in replication, it's only going to be able to make the leading strand continuously, and the lagging strand has to be made backwards in the Okazaki fragments and then glued together by DNA ligase. But also, we're only going to read this strand of DNA 3 to 5 to make an RNA, mRNA, 5 to 3. So only one side of the DNA is going to be read. And that's going to make sure that we're making the correct protein, okay? So just like with this nitrogen base, says pairing up A to T, G to C, keeps the continuity of the genetic message. Only reading one side to make my RNA also keeps the continuity of my genetic message, right? And that is it for the first review. You can email me any questions you have on this or anything you would like me to clarify. I will either get back to you, or if I have a lot, I will try to create another PowerPoint or something else that you can look at. And remember that if you have done the questions, they will be put into PowerSchool. It is not mandatory, but I strongly encourage you to do it. And if you want to see, again, how you'll be asked on the AP exam, I did open up all the multiple choice progress checks, so you could try that Unit 1 progress check if you want to, okay? So let me stop this and have a good day.